Welcome to our channel by Gulab Sir for Wisdom Coaching Center. As a part of series of lectures, today's topic is functional anatomy of nephron. As you know that nephron is a unit of excretion and it is what shown here in diagram. But better to understand by having that diagram practically. You are aware about that each nephron has a Bowman's capsule. This Bowman's capsule receives a afferent artery that brings blood in. This afferent artery further get branch into top capillaries, and this is continue to have this network through this Bowman's capsule. Now together this Bowman's capsule and these tough top capillaries is called glomerulus, and these two together form the Malpighian body. So this cup-shaped structure is a functional unit for ultrafiltration. It is formed by top capillaries and artery. That what arteriole and capillaries what from the network is a glomerulus, and this U-shaped structure, cup-shaped structure is Bowman's capsule. Together this structure is called as Malpighian body. This is continued into a short horizontally placed structure called PCT, proximal cannulated tubule. PCT, proximal cannulated tubule. This what turn inward toward the medulla region and become narrower. Later it become narrower. This narrow long tube which turn into medulla region is called loop of Henle. It is slightly broader towards the cortex region, and here it is continued with DCT. This part is known as DCT, distal cannulated tubule. This distal cannulated tubule open into collecting duct. So collecting duct receives. Now here these collecting ducts receives opening of DCTs. PCT internally lined with cuboidal ciliated epithelium. These cube-shaped cells, they are ciliated. These are the different parts what I labeled over here. Loop of Henle. This is descending loop of Henle. This is called descending loop of Henle. This is ascending loop of Henle. Now, this Bowman's capsule is internally lined with squamous epithelium. Squamous cells are flat tile shapes, and they are innervated with this cuboidal epithelium. That gap between this cuboidal epithelial cells is called microslit, and these microslits are called podocyte. These microslit, SLIT slit in this squamous epithelium are called podocytes, through which that ultrafiltration occur. Now continue to this endothelium. This capillary endothelium is also internally lined with squamous epithelium. So here also squamous epithelium, and gap between these two is called basement membrane. So three layer form in between these three parts, that endothelium of blood capillary, gap between these two basement membrane, and inner lining of Bowman's capsule are squamous epithelium with innervated cuboidal epithelium forming a slit for ultrafiltration called podocytes. Now this is continuing to PCT. Internally it is lined with ciliated cuboidal epithelium. Which cuboidal epithelium? Ciliated cuboidal epithelium. And what it continue with this? This loop of Henle is internally lined with squamous epithelium. Squamous epithelium, very thin. So here this ciliated cuboidal epithelium squamous epithelium, this is also squamous epithelium. Here again, cuboidal epithelium. And continue that cuboidal epithelium in collecting duct. So finally, the structure which form overall structure is called kidney tubule, which composed of Bowman's capsule, PCT, descending loop of Henle, ascending loop of Henle, DCT, and collecting. So here all these structures are involved in the process of urine formation. They are involved in the process of urine formation. Ultrafiltration occur in Bowman's capsule. Reabsorption continue from PCT, loop of Henle, DCT, as well as in collecting duct. Special secretion that occur in PCT and DCT, and that we are going to learn in the process of formation of urine. As a part of structure of nephron, this is what structure that you have to draw and explain, which shown in diagram also. Here you can see that structure is shown here as I have drawn. Afferent artery that enter, efferent artery that come out and form a tuft of capillaries around this loop of Henle called vasa recta. This tuft of capillaries around this is called vasa recta. Can be this is a tuft of capillaries. It is written here. Tuft of capillaries around this is called vasa recta. Collecting duct as shown in diagram. DCT, this is PCT. This immediate one is PCT, and very one that what this area, that DCT is in connection with collecting duct. Afferent artery that enter and carrying blood, same can be shown here. This is afferent artery. This is afferent artery, arteriole that bring blood into the Bowman's capsule, and this is efferent arteriole that carry blood out, and further this branch and form tuft of capillaries all around this. Here you can understand this is what network form around. This is called vasa recta. Vasa recta. Actually, this vasa recta help to maintain the osmotic concentration deep into the medullary region by having selective absorption by counter current system. That filtrate which flow in kidney tubule and blood flow in blood vessel, it is in opposite direction called vasa recta and counter current system. And this arrangement of this blood vessel around the kidney tubule is vasa recta, and movement of filter in kidney tubule and movement of blood in blood vessel is in opposite direction of the counter current system that help to maintain osmotic concentration into the medulla region. Now very important is that question can be asked that what kind of epithelium is present in this DCT, PCT, loop of Henle and DCT. So question can be asked, PCT, loop of Henle and DCT are internally lined by ABC. This tissue will be given as a part of that choice: ciliated cuboidal epithelium, squamous epithelium and cuboidal epithelium. So you please remember, as well as what are podocytes, or podocytes are located in. So point of NEET. So about this different types of epithelium and podocyte, that question can be asked in NEET. Counter current system, that blood flow in opposite direction can be defined in different way to confuse and have multiple choice question. So blood filtrate flow in descending loop of Henle and ascending loop of Henle, it is an opposite direction to the blood flow through this artery and venules, is counter current system. So this can be asked in NEET. I hope that you did understand this diagram, as it is given in book, as well as you can refer to understand the diagram. This diagram is given in textbook also. So with this explanation, I hope you did understand that structure of nephron completely. Now we shall learn in next video about the role of these different parts of the nephron in the process of urine formation. I hope you did understand this structural aspects of the nephron. Even if you are in doubt, you can ask in comment section. I appeal you to subscribe and like our channel. Also press bell icon to receive new videos as and when they are uploaded. Thank you.